And it's pretty much as I described. The first step is the first messenger, plugs in there and it opens up the channel. So, for example, this is a calcium ion channel. So when it opens up, there's high calcium on the outside, there's low calcium on the inside of the cell. And so there's going to be a huge flux of calcium on the inside. This calcium here will then go to create an electrical signal, which will then activate voltage-sensitive proteins that then will create a response. You could also have an effect where you have a G-coupled protein. When it gets bounded to, it in turn will then affect an ion channel. So there's more of an indirect way of doing it, but we still see that a lot. Once again, I'll talk about G proteins in a minute. By the way, this graph is also, it's not a sequence. When it says 1, 2, 3, 4, these are not all the steps that happen sequentially. These are possibilities that can happen within a single transduction, but I don't think the book does a very good job in telling you that. But these are the ways that an ion channel can be, or any kind of receptor, any other kind of channel protein can be activated. It can be activated directly, or it can be indirectly activated by a G protein complex. All right. So, this G protein receptor, the GPCR. So this is the signal transduction for the GPCR adenylyl cyclase signal transduction. So this is a very specific example of how a G protein is used to create a cell response. This is not the only process that G protein is involved in. G proteins can be involved in a whole bunch of different types of cell transductions. This one in particular is the adenylyl cyclase signal transduction. But they all more or less kind of work in a very similar way. So if you understand this one really well, you'll be able to understand other ones. And so the idea here is the first messenger here will bind to the GPCR, and this will activate the G protein. The G protein then activates this adenylyl cyclase, and all it does is it takes an ATP, that unit of energy, and breaks it into 3 what we call cyclic AMP. These 3 cyclic AMPs will then activate these protein kinase A's. And protein kinase are enzymes that essentially just add a phosphate group to enzymes, or other proteins. That's all they do. They take a phosphate group and plop it onto a protein. That's all a kinase does. So then these protein kinases then phosphorylate proteins, and that's what all these guys are. And then these phosphorylated proteins are either activated or inhibited. It depends on the exact process. And so whatever the phosphorylated proteins do, that will create the cell response. So once again here, we're seeing the cascade here from the cyclic AMP to the kinase to the phosphorylated proteins, and we're also seeing an amplification here, where one ATP can give rise to a whole bunch of phosphorylated proteins. I saw a question over here.
What is a kinase again? What does it do?
Kinase basically it phosphorylates a protein. It adds a phosphate group. And by the way, the G protein is called G protein because it uses, instead of ATP, it uses GTP. So it's using another nucleotide. It's called instead of adenine triphosphate, it's guanylyl triphosphate, and basically it's playing around with the phosphorylation of GTP to GDP. You don't really need to know that in super detail, but that's where the G part comes from the GTP it uses. All right. So receptor enzymes, like I said, pretty straightforward. The first messenger binds onto the receptor. This receptor then activates, sometimes inhibits, an enzyme. In this case, it's this enzyme called tyrosine kinase. So it's the tyrosine kinase because it uses it's built up of the amino acid tyrosine, and it's a kinase, so it's it's adding phosphate group. So it's taking this protein, taking some ATP, and whatever phosphate group that comes off the ATP is being added to the protein to make a phosphorylated protein. So this is an example of a receptor enzyme, and this is once again a catalytic receptor. Integrins work something pretty similar. I don't really need you to know the details of it, but just it works pretty similar, but it just interacts with the cytoskeleton. All right, so your book has this huge figure that summarizes all the possibilities of signal transduction. I think it's a good way to review it. This is, I think, figure 6.10. After you read through all this part, if you haven't done so already, go back and look through this. See if you can understand each step. And once again, remember, this is not the full sequence. This is all the possibilities that can happen during signal transduction. The idea is just to get from point A here, from the signal molecule, down to here, point B, to a response. And the way you go about it can vary, right? There's a whole bunch of ways you can get there, and it's going to vary based on the specific process and the specific signal. That's the main punchline here. I want you to know the one specific example, but know that it can be abstracted to other processes. We good over here? Any questions? Making sense so far? Yes.
What's the voltage gated channel?
Voltage gated channels. So we talked about them. We talked about transport of proteins. Those are channels that will open and close based off the cell's voltage. So that's electrical kind of potential. We'll talk actually more about that next week. But yeah, that's basically it is. There's two other types of gated channels. You can have mechanical gated, so if they feel some kind of stretch, they'll open or close. And you can have chemical ones, which this is an example of a chemical gated channel. It gets the chemical a ligand. When it binds to it, it opens or closes. The voltage is similar in the sense that it detects the voltage difference from the inside and outside of the cell, and then it will open or close depending on that voltage.
So it doesn't necessarily be the ligand.
No. Mechanical and voltage gated channels do not necessarily need a ligand. The chemical gated ones need a ligand.
So the ions is the chemical.
Well,
But
no.
the ions
The first messenger or some other ligand intracellularly is the ligand.
What?
So let's look at this pathway. So signal molecule binds to gated ion channel. This is the first messenger, right? And since it's binding directly to the channel, this is a chemical gated channel. Because it's opening and closing based off this ligand, which is a chemical, it's a compound, right? And then this one really lets in ions. So it's an ion channel, but it's chemically gated.
Okay, got it.
Now you can get in these weird territories where, and we'll talk about this next week, where you have a voltage gated ion channel that lets in ions. So if it gets activated and it lets ions in, it's going to change the voltage, which could affect whether it's open or closed. And you have this weird kind of feedback loop, which is confusing, and we'll talk about that next week. So I'll question this area. No questions? Okay. All right, notable signaling molecules. A big one is calcium. I don't need you to right now need to know all the different things calcium is used for, but just know it's a prominent signaling molecule. Calcium is generally in high concentration outside the cell, or sometimes in compartments within the cell, like in the endoplasmic reticulum, and then they get released and they're used as a signaling molecule, usually as a second messenger or some other thing to activate another second messenger. There are also, the book goes on a whole bunch of, I talk about a whole bunch of other molecules. I just want you to know that gases can be a signaling molecule, and icosanoids, it's a type of lipid. the lipid-like structure. It's kind of related to cholesterol, but it's different. And here are the different gases. And nitrous oxide was a big discovery recently. And carbon monoxide, hydrogen sulfide. You don't need to know exactly what they're doing and how they're doing it. You just gotta know that these can be potentially used as signaling molecules. Alright, receptor modulations. So your receptor can be, affected. It can be change how it behaves. It's a protein. So all the ways you can affect a protein, all the properties a protein can, can experience, are the same thing for receptors. They can experience saturation. They have different varying amounts of specificity. They can have competition for their binding site. We can look at this example here. This is the first messenger here. It binds and it eventually creates a response. But you can have molecules called agonists. Agonists act a lot like the first messenger, to the point where they can bind and even create a response. And so, a common example of an agonist is basically many drugs out there. Drugs are put out there so they can mimic these primary ligands and promote this effect. An example of an agonist for a receptor is nicotine. There's nicotinic receptors there, and they bind to various endogenous molecules. Endogenous meaning they're made on the inside of the body. But nicotine is an external factor that's an agonist because it can also affect those receptors, those nicotinic receptors. And then you can have antagonist receptors that basically block the receptor, meaning you're going to get no response.